Welcome everyone, and thank you for joining us. Today we are honored to have Dr. Douglas Boreham, Dr. Christopher Thome, and from inside SNOLAB, Dr. Christine LaLonde, presenting SNOLAB, researching the effects and the presence and absence of ionizing radiation repair project. My name is Cassandra Carlson, and I will be hosting the webinar in the back end. This webinar is approximately 45 minutes, followed by a Q&A session. Moderating for us today is Dr. Mark Mendonca. Dr. Mark S. Mendonca is a professor of radiation oncology and medical and molecular genetics and director of radiation and cancer biology at Indiana University School of Medicine. Dr. Mendonca has extensive expertise in both X-ray and proton radiation biology. His research is focused on increasing the effectiveness of radiation in lung and pancreatic cancer treatment by biomedical and genetic mechanisms, as well as physical approaches, protons, nanoparticles, and flash. Since 2011, Dr. Mendonca has served as the editor-in-chief of the Journal of Radiation Research. In April of 2020, Dr. Mendonca was appointed Associate Vice Chancellor for Research at IUPUI. And I'll hand it over to Dr. Mendonca to introduce Dr. Douglas Boreham, Dr. Christopher Thome, and Dr. Christina LaLonde.
Thank you, Cassandra. It's my pleasure to introduce our presenters today. Dr. Douglas Boreham is currently a professor at the Northern Ontario School of Medicine and is division head for the medical sciences. Dr. Boreham is a recognized leader in the field of low-dose radiation health and environmental effects. He was selected as an expert Canadian delegate for the United Nations Scientific Committee on the effects of atomic radiation. He's earned a number of awards, including the McMaster President's Award for Excellence in Instruction, the Canadian Nuclear Achievement Award for Outstanding Education and Communications, Canadian Radiation Protection Association Award, Distinguished Award, recognition of outstanding contributions in the field of radiation protection, and importantly, also a Mentor of the Year Award by the Radiation Research Society. Dr. Chris Thome is an assistant professor in medical sciences at the Northern Ontario School of Medicine. He holds an industrial research chair position supported by Nuclear Innovation Institute. Dr. Thome's background is in radiation sciences. He earned a master's degree in health and radiation physics and a PhD in medical physics from McMaster University. His current research interests are low-dose radiation biology, specifically focusing on radiation carcinogenesis, the biological effect of subnatural background exposure, and radiation-induced cataracts. Dr. Christine LaLonde is from Newfoundland, Canada, and joined the radiation biology lab at NOSM in 2016. When she completed her multidisciplinary PhD in biomedical sciences, her research focused on behavioral neuroscience, specifically fetal programming of behavior in offspring of low-dose exposed animals. Moving forward in a similar vein, her postdoctoral research utilizes a small nematode C. elegans to investigate epigenetics. Using SNOLAB for sub-background radiation environment, the current project is focused on phenotypical changes in behavior, reproduction rates, and neurological function. So first up, I'd like to say hello and hi to Dr. Doug Boreham, and Doug will begin his presentation. Thank you.
Well, thanks for that kind introduction, and thanks for inviting us to give our lecture today on the SNOLAB. I would like to start off first by explaining the background slide that I have showing a shark with a laser on his head. Several years ago, when we were at the radiation research meeting, we discussed the idea of having a secret underground layer lab where we would study the effects of low background radiation. And of course, we brought up the slide of Dr. Evil in his lab with sharks with lasers on their head. And ironically, several months later, a package showed up in the mail and it had a shark with a laser on his head. So this has been our model. And so we are going to take you today down into our secret lab called the SNOLAB. I will share my screen now. So we have 3 speakers today working in our lab. And of course, it's called Researching the Effects and Presence of Absence of Ionizing Radiation, or the REPAIR project. I want to start off by explaining what NOSM is. NOSM is the Northern Ontario School of Medicine. And you can see that in the left-hand corner, our model has something in English, something in French, and something in Ojibwe. So we do a lot of Francophone and First Nations research and work at our schools. We have 2 medical schools, one on East Campus in Sudbury, and we have the West Campus in Thunder Bay. Our sister universities are Laurentian University and Lakehead University. And of course, we have these labs have state-of-the-art research laboratories for cell biology, molecular biology, Each of the campuses have an animal facility with X-ray machines. So we're well established as a radiation biology group. And of course, we have the SNOLAB, which we'll talk about today. The Sudbury campus is on the right, and that's the Great Lakes you're looking at where the arrows are pointing. Lake Huron, the north shore of Lake Huron is where Sudbury is, and the far end of Lake Superior is where the Lakehead campus is. So we're in Northern Ontario, and that's why we're called the Northern Ontario School of Medicine. And like I said, we have a social accountability mandate to provide excellent research and health for the people of Northern Ontario. All right, so let's start a little bit about, before we get going, here's the campus in Sudbury, and you can see a picture of this little electric car that we have as part of our research infrastructure. The car was donated by Bruce Power, and we also have a little logo of the SNOLAB above the back tire there. This car is used to transport our researchers from the campus to the SNOLAB. Now, the SNOLAB is in a mine sort of out in the bush, about 50 kilometers away or about 30 miles. So each day this car can be used by our researchers to go out there. Because it's electric and it's in Ontario, we can call this a nuclear powered car, because in Ontario, we are about 60 to 65% nuclear powered after we phased out coal in 2014. So we got a little nuclear powered car to take us to the SNOLAB. It is a Ford Focus. I did try and get Elon Musk to give us a Tesla, but he never answered my email. So there you go. So here's the SNOLAB. Here's some pictures of the SNOLAB. The top left corner with the big sphere is the neutrino detector in the SNOLAB. The neutrino detector was used to look at the differences between the types of neutrinos that come from the sun during the fusion reactions. And it was the main focus of the lab when it was built. Since that time, astrophysics has gone really ballistic in the SNOLAB, and there's a number of instruments down there that are world-class and state-of-the-art. The neutrino detector on the top left is just showing the guys constructing it before area was filled with water. Now that detector had something like $200 million worth of heavy water in it and thousands of photomultiplier tubes that were used to detect the different types of neutrinos that entered the lab. The picture on the right at the top is what we call HALO, and that is a supernova detector for astrophysics. And when I was in there once, there was a student sitting there working on it. It was part of their project. And I asked them, how many times does supernova happen? And they said, once every century. So I just looked at it and said, I hope you're feeling lucky today, because if it's part of your thesis, you've got to have a bit of time to wait. The bottom left-hand picture is a dark matter detector. There's several types of dark matter detectors down there because the challenges of finding dark matter are immense. So there's several different designs of dark matter detectors, and that's one of them there that you see at the bottom. And of course, now we have on the right-hand side at the bottom is our life sciences lab that we call Life Sciences and Chemistry Lab, and it's been established by a lot of hard work by our graduate students and Dr. Thome and our postdocs. So it's been quite a challenge, and as we get through this discussion, Dr. LaLonde is down in that lab right now, coming to us live as we go through this talk. The SNOLAB, what used to be called the Sudbury Neutrino Observatory, the new name now is just called SNOLAB, but the SNOLAB was instrumental in allowing Dr. Arthur McDonald to win the Nobel Prize in Physics in 2015, because he was able to discern the difference between a special type of neutrino that physicists could not figure out on the surface of the Earth. So they had to have some 2 kilometers of norite rock shielding to shield out most of the cosmic radiation hitting the Earth's surface, so that they could work out what that third type of neutrino was. And I am not an astrophysicist, so I'm not going to go there. We were fortunate enough to have radiation research produce a SNOLAB focus issue, and that focus issue is there, volume 188, for anyone if they weren't interested in looking at it. But there's a number of really interesting articles in there, including a commentary forward by the Dr. McDonald, the Nobel Prize winner, several reviews, and some of our original work on looking at low doses of radiation and how low doses of radiation have various types of stimulatory effects on organisms. And I think the reason that we are in the SNOLAB is because we have a hypothesis that with our experiments to show low doses stimulate biological processes, in the SNOLAB, in the absence of all this low dose radiation that we're exposed to, hypothesizing that in fact that may not be great for life, and it may be detrimental to life processes in the process of evolution. All right, so let's just talk real quick about the SNOLAB. This picture on the right-hand side is a diagram of what what the SNOLAB is designed like. It's in, like I said, the Creighton mine, about 30 miles from Nossum. If you look at the picture on the right, the surface is the blue, and the vertical black lines going down represent mine shafts, which basically take down cages or elevators, if you want to call them, down to the various ore bodies, which are shown here in yellow. And the ore bodies are mined and brought to the surface on the skips. They're sort of the opposite of the elevator. And each of these vertical lines go down further and further. And you can notice that shaft number 9 is the one that goes down to the 6,800 level. And that is the level that we get off to get to the SNOLAB, which is 6,800 feet or roughly 2 kilometers underground. And you can see that that norite rock is above that 6,800 level. And it's equivalent roughly to about 6 kilometers of water shielding. So that is a lot of shielding for shielding out the various types of cosmic radiation. The lab itself is roughly 33,000 square feet. And the lab that we have is roughly somewhere between 500 and 700. I'm going to say 700 just to make us feel more important. If you look down at the very bottom of the slide, we see the 6,800 level. The SNOLAB is to the right. That represents a walk down a mine drift of approximately 1 mile to get there. And you're wearing full miner's gear, and you walk to the SNOLAB and enter this class 2000 lab that you then start doing your research with. So the SNOLAB compared to the surface is roughly 50 million times less cosmic radiation. The slide on the right, you'll see that there's coating on the wall. It looks very smooth. That's a polymer coating that reduces radioactivity, keeps radon gas out, and other things like that. And if you look at the center slide, you'll see a number of our researchers walking down that 1 mile walk from where the cage enters the 6,800 level and where the SNOLAB is. So you're wearing a miner's lamp and you're walking down this tunnel, and it can be very daunting to some people. On the left is the actual mine that you look at where the cage is launched at the surface. So we're going to talk today about the repair project, and what you see is a diagram of the intricate tunnel system that represents the underground SNOLAB, like I said, 6,800 feet underground. The repair project is that little circle where the arrow is. And at this point, I will turn it over to Dr. Thome, who will tell you exactly what we're doing down there and what our hypothesis is.
So before I get into you talking a little bit more about our specific project, I'm just going to go through a little bit of data that's already been collected from other groups. So this field of kind of subnatural background radiation, it's a pretty niche field, but there have been a few other groups that have looked at what happens when we bring various different model systems into these sub-background environments, and what does that do to their growth and other processes. And so what they found can kind of be grouped into 2 different findings. And so the first is related to the growth of these organisms. And what they found is that if you remove natural background radiation, that these different systems it impairs their growth. And so their growth rate decreases. Early studies that were done back in the 1970s focused on paramecium. And so these ones weren't actually done in underground labs like we're doing. They were done more just using some kind of artificial shielding on the surface. And so they were only able to get kind of a very modest decrease in background dose rates, but nonetheless, they still saw these findings. And so when you subject these systems to a sub-background environment, you get an impairment in growth. And so this has been seen in a couple of different systems, bacterial species like blue-green algae and Deinococcus radiodurans, also with yeast. And more recently, some groups have looked at mammalian cell lines. Most of this has just been done with the very basic model systems, although work is now moving into some more kind of complex multicellular systems. And one of the ways that they kind of prove that this is due to that sub-background radiation is that when you artificially restore that background dose rate, so you take something like a low activity cesium-137 source, when you introduce that into that sub-background environment to artificially reintroduce natural background radiation, those growth rates are restored back to normal levels. And the other thing that these groups found is that when you remove natural background radiation, what it does is it impairs the abilities of these systems and these cells to repair genomic damage. And so we see an increase in damage and a reduced repair capacity. And so this has been shown through a couple of different endpoints. So survival fraction is reduced. They found an increase in background as well as induced mutation rates, an increase in micronuclei formation, and an increase, reduced ability to scavenge free radicals. And so as Doug kind of mentioned, what the hypothesis is that this kind of low background level of radiation helps to keep some of these natural processes, the kind of repair and antioxidant systems upregulated to a certain level. And so when you bring these systems to this sub-background environment, over time, they're going to adapt to that sub-background environment and will start to downregulate some of those systems so that now we start to see this increase in genomic damage. And if you challenge these systems with a high dose of radiation, we see higher rates of induced mutations and a decrease in survival rates. We still don't know a lot about how this actually happens, and so this is what we're exploring down in SNOLAB. So in addition to SNOLAB, there are a few other underground labs around the world. And like SNOLAB, a lot of them started as an astroparticle physics lab, and they've kind of now moved into doing some sub-background biology work. So here's SNOLAB where we are. The two other main ones right now that are doing sub-background experiments are LNGS here, which is the Gran Sasso lab in Italy. And so their lab is tunneled into the side of a mountain. And then the other main one that's operating right now is WIP. And so this is the Deep Geological Repository in the US. And so they're doing similar kinds of experiments to what we're doing in SNOLAB, where they have systems underground in that sub-background environment, and they're looking at things like growth rates and genomic damage. But in SNOLAB, we are significantly lower compared to these other background labs. And so I'll talk a little bit about the kind of dosimetry we've done, but we have lower dose rates compared to what they have in some of these other facilities. So in SNOLAB, as part of the repair project, we're looking at 3 different experimental environments. And so the first one is our surface control lab. And so this is our lab at the medical school. And so our different systems that we're growing in that surface control lab, they're being exposed to that normal background radiation levels that we get on Earth's surface. We also have our sub-background environment. And so this is down within SNOLAB. And we had to design this kind of specialized tissue culture incubator, because when you go underground, we basically get rid of all cosmic radiation. And so the cosmic radiation levels in SNOLAB are about 50 million times less than you get on the surface. But we still do have some residual background radiation levels in SNOLAB, mostly from radon, and then also a little bit of gamma radiation from decay of some radioisotopes in the rock. And so we had to design this specialized tissue culture incubator, which I'll talk about on the next slide. And so that allowed us to further reduce things like radon gas and gamma rays. We also have an underground control. And so this is just a standard tissue culture incubator that we have in SNOLAB. And so the reason we have a second control is that one thing we could not control for in the surface versus underground were the pressure differences. And so when you go underground, there's an increase in air pressure of up to about 25%. And so we explored different ways that we could maybe design some pressure vessels underground or on the surface to try and kind of match those levels, but it wasn't really going to be feasible with how we want to culture our systems. So we have this underground control, which is exposed to the higher levels of radon gas and higher gamma rays underground. So it's not really that sub-background environment, but we can use it as a control for the higher pressure underground compared to the surface. And we also can use that as a control for when we have to transport stuff up and down from the lab. So any kind of influence that might have on our model systems, we have this underground control to account for that. So we're right now culturing systems in all 3 of these environments. And as I'll show in a few slides, our background radiation levels within the surface control and the underground control are pretty similar. And we have quite a reduction in our sub-background environment. So this is that specialized tissue culture incubator that I was talking about. And so what it is it's kind of a custom designed glove box that has within it a tissue culture incubator. And so on the left-hand side here, you can see we have an interlock system. And so this allows us to bring samples in and out of the incubator. We have a radon monitor here that continually monitors radon levels within the chamber just to make sure that they're maintained at a low level. We have the incubator component of it, which is designed from a lead shielding. So it's a lead castle. There's 10 centimeters of lead all around it. And so that lead helps us to reduce the background gamma rays. Inside this lead castle, we have gases being fed in that helps us maintain the appropriate atmospheric concentrations. We have a heater that gives us that 37 degrees Celsius for normal tissue culture. We maintain that 5% CO2 level. And so this whole chamber is fed with compressed gas cylinders that we know have very low levels of radon gas. So we know that all of the air entering into this chamber is going to be almost radon free. And so we have on the right-hand side here, we have control of all the different gas levels. And we also have a backup power system just in case, because we're in an active mine, there are fluctuations in electricity. We do have some power outages. And so we have to account for that. So the group at SNOLAB helped us design this specialized tissue culture incubator. And they've done a lot of this work. A lot of their detectors require super low levels of background radiation. And so they know what types of building materials to use that have low rates of radon emanation. And so we spent a couple of years building this, but it's now up and running and we have cells currently growing in there. So I said radon gas is kind of the main thing we had to account for underground. And so underground radon levels, they're not super high because SNOLAB has a pretty big air filtration system. And so there are a lot of air turnovers underground. but the levels are still higher than we are suitable for us to do these sub-background experiments. So underground, which is the blue line on this graph, they're about 120 becquerels per cubic meter. On surface, which is in green, so this is our measurements that we took at our lab at the medical school, it's about 3 to 4 becquerels per cubic meter. And so because radon has a pretty short half-life of 3.8 days, if we fill compressed gas cylinders with air from the surface, which already has pretty low levels of radon, and if we let them sit for about a month or so to go through multiple half-lives, then we'll get to the point where that air is essentially radon-free. That's what we're feeding into that specialized tissue culture incubator. And so we have levels of radon in there that are over 100 times less than we have in our laboratory air. And then the other thing we had to account for is gamma rays. So these two spectrums on the bottom here show the gamma ray spectrum underground, at the surface, and then within our tissue culture incubator. So on the surface, we get a lot of really high energy gamma rays as part of our cosmic radiation. So when you go underground, we don't get a lot of those high energy gamma rays, which is good because they're harder to shield from. But we do still have a significant number of low energy gamma rays. And so underground, these gamma rays come from decay of natural radioisotopes. So certain elements in the uranium and thorium decay chains will emit gamma rays. And so we designed, again, this kind of lead shielding, this lead castle, which you can see on the top there. And we have our tissue culture flasks incubating within that lead castle. So that 10 centimeters of lead gets rid of almost all of those gamma rays underground. And so on the right-hand graph here, which is looking just kind of at that low energy range of our gamma spectrum, you can see that we just get a small number of gamma rays between about 1 to 1.5 MeV range, but we're a couple orders of magnitude below what we have within that atmospheric air within SNOLAB. And so what we did before we really started these experiments was look at the background radiation dosimetry. And so because we knew that radiation levels within that tissue culture incubator were going to be extremely low, we couldn't really measure those levels with your kind of standard dosimeters like TLDs, because in order to accumulate enough dose on those dosimeters, we would need to leave them in there for many months or even years at a time. So our background dosimetry, we did that through Monte Carlo simulations, and we took flux measurements or air concentrations of 6 different radiation sources. So radon, gamma radiation, neutrons, muons, and then potassium-40 and carbon-14, which are within our cell culture media. And we ran the dosimetry on all those different components. And so this graph on the left here shows our background radiation dose rate in all 3 of our environments. And this is showing just from the external sources. So our radon gas, muons, neutrons, gammas. And so our surface and underground control have very similar equivalent dose rates. Different contributions though in terms of where that dose rate comes from. And so this is on a log scale, so these bars don't really show what the contribution is, but for your surface control, most of our background dose rate comes from muons, and then a little bit from neutrons and gammas. Whereas underground, our underground control, most of our dose comes from that high radon level, and then a little bit of gamma radiation. Within our STCI, that sub-background environment, we get over 100-fold decrease in our background dose rate. Most of that dose just comes from a little bit of residual radon that's being emitted from some of those building materials within the chamber. Now, I mentioned internal sources too, and so we've measured levels of carbon-14 and potassium-40 within the culture media that we're using to grow our systems. Carbon-14 levels are pretty low, so we don't need to worry too much about them. But the potassium-40 levels are pretty high. And so if we grow our cells in normal media, we do get a significant dose from that potassium-40. And so we're working right now on developing ways that we can culture these systems in pure potassium-39 to eliminate that K40 as a variable. Okay, so we're working with currently 4 different model systems. I'm going to talk about the first 2 here, lake whitefish and human cell culture. Then I'm going to turn it over to Christine, and she's going to talk about the C. elegans work that she's doing. And then we'll turn it back over to Doug, and he's going to go through the yeast work that we're doing. So lake whitefish was the first species that we started working with. It's kind of a unique species that not a lot of people work with. And so we've been working with lake whitefish embryonic development for about 10 years now. And this all kind of started as part of an environmental assessment we were doing on different environmental stressors. And we discovered that embryonic development in lake whitefish is actually a pretty good system to look at the effects of low dose chronic exposure to ionizing radiation. The reasons for that are a couple. The main one being that these species develop at a really low rate if you keep them at a cold temperature. So they like a temperature of anywhere from about 3 to 5 degrees Celsius. And at those temperatures, their embryonic development period lasts about 200 days. So we can get over 6 months of growth if we keep them at these low temperatures. And so if you compare that to a species like zebrafish, which is a much more common aquatic model, which have a development period of only a couple of days, these lake whitefish embryos that develop over this protracted period, we can look at the effects of really low chronic exposures to radiation, or in our case, to look at the effects of potentially bringing them in a sub-background environment. They're also really low maintenance, which is good because we have kind of intermittent access to SNOLAB. Depending on what happens in the mine, sometimes we can be locked out for a couple of days at a time. So they're really low maintenance. We grow them in Petri dishes with water in a fridge. You change the water out maybe once a week. And so they're pretty conducive to running experiments in a lab like SNOLAB. So we did some experiments on lake whitefish back in 2015. This is what the lab looked like back at that time. So this is before we kind of set up this whole life sciences lab within SNOLAB. And so at that point, it was kind of just a storage hallway. We brought down a couple of fridges that we set to 3 degrees and 5 degrees, and we grew some embryos down there for a 6-month period. And really what we were looking at for this study was just what is the feasibility of running a full biological experiment down in SNOLAB, because no one had ever done that before. And so this was really just kind of a pilot study. We didn't control for radon levels or the gamma rays at this point. We really just wanted to know, can we run a prolonged experiment down in SNOLAB with people coming up and down the cage, bringing supplies back and forth. And so we were able to successfully do that for their whole development period for that 6 months. So what we did is we collected lake whitefish. And so how we get these embryos is we go out on Lake Huron in the middle of November. We catch spawning adult male and female fish. You strip the eggs and the milt, and we do in vitro fertilization. And then we brought these embryos back to Sudbury. We sent some of them down to SNOLAB. We kept some of them on our surface lab. And we kept them at the same temperature underground and on the surface. And we just monitored them for that 6-month development period. And then we sampled them at several different time points to look at size and development rate. And so what we found, this is showing the body mass and body length of the embryos as they're at different points in development. And so as we go from 40 to 60 and 80% development, we see an increase in body mass and body length. But what we did see is that we do have some significant differences in growth underground versus on the surface. And we actually see that underground, there's a slight increase in growth and size compared to what we get on the surface. And now again, we can't really relate this back at this point to radiation levels because we didn't in this case control for the things like radon and the gamma rays underground. But this kind of just shows that it is possible to run an experiment in SNOLAB. And we do see some significant differences when we culture things underground versus on the surface. So this was really just kind of the first pilot experiment we ran. Where most of our work is now currently is looking at human cell culture systems. And one of the cell lines we're working with is the CGL1 cell line. And so this is part of the collaboration that we have with Mark and Donka. And so I think at that same Radiation Research Society meeting that Doug was talking about, we started chatting with Mark about the SNOLAB project, and then he was interested and came up to Sudbury. We brought him on a tour down of SNOLAB, and he introduced us to the CGL1 model system, which is a system that he's done a significant amount of work on. So essentially what this system is it's a hybrid cell line of a malignant HeLa cell and a normal human fibroblast. And so when they created these hybrids, there was a number of different segregants that were produced, but one of them, the main one, is the CGL1 cell line. And so the CGL1 cell line is a non-tumorigenic cell when you inject it into animals, but when you grow them in culture, they do have a low spontaneous rate of cell transformation in culture. And they're also very sensitive to low doses of stress. And so when you add or when you add radiation or other kind of stressors, we do see changes in that background rate of spontaneous transformation. And so they're a good system for looking at subtle changes in environmental stressors. And so we thought they would be a good model system to look at down in SNOLAB. And so over the past few years, we've done some work with this model system. And this is a study that was done by Jake Perkinen, who's one of our postdoctoral fellows on repair. And so this was kind of just some preliminary work getting ready for what we're going to do down in SNOLAB. But what he did is a full transcriptomic study on the different variants of the CGL1 system to look at what genes are regulated, up and down regulated in the different variants to try and understand the difference in the transformation rates that we see between these variants. And so this is kind of some preliminary work that we did where we're going now, but we're going to grow these cells underground in SNOLAB and look at different factors in these cells. So this is just a graph from a study that was done by Redpath back in 2003 at UC Irvine. And so this is looking at transformation rate or transformation frequency in these CGL1 cells. And this was looking at the effects of low-dose diagnostic X-rays. But this is kind of one of the reasons why we wanted to use the cell line down in SNOLAB. And so you can see at the dose of 0 gray, there is that baseline rate of cell transformation, of neoplastic transformation. And you can quantify that just by counting the colonies that form within these culture flasks. And what they found is that if you increase the radiation dose, if you give a little bit of radiation, and we're talking in fractions of a centigray, you actually get this suppression in background transformation rate. And when you get to higher doses, then you get an increase. And so they saw this kind of J-shaped curve. Now, this 0 gray represents normal background radiation. And so what we're hypothesizing and what we're looking at right now is if we remove background radiation, so if we kind of go on the other side of this axis here, we're hypothesizing that we might actually see an increase in transformation rate based on what other groups have seen. And if you just kind of follow the shape of this curve, we might actually see that bringing these cells underground, exposing them to that sub-background environment might actually be detrimental and lead to an increase in transformation. So this is one of the endpoints we're looking at right now. So currently the experiment's in progress. We are running things over a fairly prolonged period of time because we think we're talking about some kind of changes that are going to happen in terms of expression levels of different factors within these cells. We think this might take a more prolonged period of time to see some effects. And so we are going to grow cells underground for up to 6 months, and we are sampling them at several different time points. And what we're going to look at is in these sub-background adapted cells, do we see any kind of baseline changes in different endpoints? And also, do we see a difference when we expose these to a high dose radiation challenge? And so if we take these sub-background adapted cells, and if we hit them now with a high dose of radiation, do we see an increase in damage and a reduction in survival? And so we're looking at things like more basic endpoints like growth and survival. We're also looking into genomic damage, oxidative stress, transformation frequency, and then we're also doing multiple omic studies on these cells. So this is kind of where we are right now with the human cell culture work in the middle of these kind of prolonged experiments. So with that, I'm going to turn it over to Dr. LaLonde, and she's going to give us a little tour of SNOLAB and then talk about some of her C. elegans work.
Hello, everyone. My speech will not be terribly long as this laboratory is quite noisy and fairly dynamic in activity. As mentioned by Dr. Thome and Dr. Boreham, my research focuses on a small nematode model, which is a 1 millimeter long C. elegans, which is a transparent worm that's thousands of organisms compared to human genome. Model is perfect for the ground, and it can reproduce an entire generation of offspring within 3 days. So we can look at multiple generations of offspring. Conditions. So this little nematode moves fairly fast. So this is a still photo. There's only one in the photo. The rest of the little squiggly lines are actually tracks from where the worm has already crossed across the agar. It produces about 100 to 200 eggs within an hour, and those eggs will grow up to be full adults within 3 days. So for the function of epigenetics and reproduction and neural development, it's an ideal model to work with underground. Currently, the underground surface worms are living in this small little incubator at which I can keep standardized at room temperature. And today, some new C. elegans have come underground and are about to enter into the STPI for the sub-background radiation cohort. And they will go outside. I've opened up the doors so that people can see where the cells live. However, the worms live at room temperature, and they're going to go off into the corner underneath where the door lives. Functionality-wise, we look at motor function within the worms,
And we look at short and long-term memory, which are easy assays to reproduce above ground and underground. So thank you. And Dr. Boreham, I'll return it to you.
Well, thanks, Christine. That was great. The audio was a little bit off there a bit, but the bottom line is we're very excited to have this new model system down there. It's just been a recent introduction to our facility, and Christine's been working on it for several weeks underground. So we're really excited to find out what's going to happen with this very nice system to study background radiation. All right, I want to talk to you now about a collaboration we are doing with NASA Ames Research Center in California. We were fortunate to team up with them on a project that they're doing with studying biological systems in deep space when we travel into space. So I want to talk to you a little bit about that project. Their project is called the BioSentinel mission. And the collaboration between us and them is going to be very interesting because they have a little spaceship where they're going to put living cells on it. And that object you see on the right-hand side is called the CubeSat. And this will be launched into deep space, some 40 million kilometers in deep space. And living organisms will be monitored to understand the effects of the types and quality of radiation in space and how that impacts DNA and how cells can respond and repair to that. So we have what we call autonomous biosensors in deep space. And the important part about those biosensors is that they need to be able to survive these living cells for extended periods of time with very minimal life support technology. And second of all, they need to function reliably with intermittent communications with Earth because, of course, they're going to be moving deeper and deeper into space as they go. So this is a little sort of caption that was advertised a few years back where NASA is sending some lucky yeast into radiation-filled deep space where no yeast has gone before. And of course, when we saw that, we thought this would be a great opportunity for us to collaborate and start understanding the difference between radiation in space or the absence of space radiation. Now, the system is called Saccharomyces cerevisiae. It's the yeast system, and Sarco stands for sugar, and Myces stands for fungus, and cerevisiae stands for beer. So basically, we're sending a sugar fungus that makes beer into deep space. We're also going to be bringing that down into the SNOLAB to understand the opposite effects of cosmic radiation. Now, these yeast actually make a very suitable astronaut because for a number of reasons. They're living, they need no air, they need no water, they need no food, they need no light, and they're alive. And basically with minimal temperature control, we can actually have these bio-living organisms accumulate space radiation in their DNA as they move through space and then figure out a way to look at what that damage has caused. Now, the beauty of these organisms is that they're a eukaryotic cell, similar to mammalian cells. They're highly studied and understood. Their genome has been sequenced. Their genetics are easily manipulated. They're very easy to maintain in the lab in space. And of course, they make wine and beer and can make bread out of them. So I guess if we're going to go to deep space, that's probably an option to have. All right, so our collaboration, like I said, is that we want to understand the effects of ionizing radiation in deep space as well as deep underground in the absence of that radiation that we're talking about. The methodology involves several yeast strains. The Saccharomyces wild type yeast strain is called YBS71-1, and that's the NASA wild type yeast. And wild type means it's normal with respect to all its DNA repair process and other metabolic processes. And then there is a RAD51 genetic knockout yeast strain, which RAD stands for radiation sensitive. And the RAD51 yeast cannot do very good double-strand break repair. So consequently, that double-strand break repair is inhibited in these yeast strains. So initially, we had to sort of figure out a way to do the actual experiment. And when the yeast are desiccated, this graph shows the desiccated yeast starting at day one. So the two strains of yeast, the top line represents a wild type strain, the bottom line represents the double-strand break repair deficient strain. And when the yeast are desiccated using a certain methodology, they are then they're rehydrated and brought back to life at various times. And you can see on the bottom of the graph, it's time in weeks. It's hard to see the actual weeks that they're listed there, but the wild type yeast clearly are able to recover from desiccation at a higher survival rate than, of course, the RAD52 or the RAD51 yeast is the lower line. And you can see that they've gone out quite a long ways in time, 80 weeks, 90 weeks, looking at how the yeast can recover just on the surface of the Earth. And of course, when these things are in space, they're going to be accumulating damage from various types of high energy, high LET types of radiation quality, and their recovery will be indicative of how their DNA was damaged on the trip. Now, what we're doing is we're replicating this exact experiment, and we're involving understanding the stress response processes in these yeast cells, and also how transcriptomics and proteomics can help us understand what is happening to these yeast with the radiation qualities that they've been exposed to. So just in the BioSentinel system, that little spaceship I showed you earlier on, we'll have these multi-plates you can see on the bottom here. And the plate on the left represents the RAD52 strain, which is a double-strand break repair deficient strain, so very radiation sensitive. And when these cells that are desiccated are rehydrated using a robotic system in the little spaceship, they are rehydrated with nutrient media, and it will start to metabolize and recover from whatever damage has happened when they've been traveling. Now that one on the left is at time 0, so clearly there's nothing metabolizing, because when the cells start to grow and metabolize, they will change the color of the media using a metabolic marker. And you can see in the middle graph, or the middle plate, this is after 19 hours, you can see that the first vertical row is 0 dose, and you can see 3 of the wells are pink, which means they have live cells in them, and the control with no cells in the bottom is the blue one. And then as the dose increases, you can see that after 19 hours, at 30 gray, because these are repair deficient yeast, we're not getting a big recovery there. And then after 24 hours, you can see that there's a little bit more recovery, but still after 30 gray, these radiation sensitive yeast aren't recovering as well. And the quantitation of the color of these wells will be used as yeast cells travel deeper into space to see how long they can recover from that process of being irradiated with cosmic rays. So, of course, NASA has their crest and their stickers called the BioSentinel Ames Research Project. And we have our repair sticker now. One of our students was very creative. And instead of having a yeast in space with a space helmet on in a little spaceship, we have now our yeast underground going down a mine shaft with cosmic rays at the green on the top. And he's got a miner's lamp, and this yeast is going to be down there. And we're going to try to understand the whole process of shielding living organisms from space radiation. So up to now, we've been able to reproduce what NASA has done with their yeast strains in terms of survival after desiccation. The lines you see on the graph represent the 2 strains that NASA has. And we have another strain that we're looking at that has a slightly different ability to respond to stresses. And you can see that we started at day 0 with 100% of the yeast recovering. However, as time goes on, you can see that, in fact, we're getting less viability after the recovery. And you can see we started this in September of 2019, and this graph ends at March 2020. And the reason it ends is because that's when COVID hit, and that's when they shut our labs down. And when you're doing a long-term experiment like this, it's very difficult to, it's a challenge. So we lost that whole sort of experiment. So we got to start over again. And we do have a graduate student who's just started, and he will be reproducing this data, and we'll try and start again. Hopefully there won't be another pandemic coming up. But clearly our results are very similar, and the bottom line, the RAD51 strain is not doing as well as the wild type strain. And so we're learning a lot about what's going on in these cells. And I mentioned before that this BY4743 cell, the blue line, has a different response to stress than the 2 NASA strains we're working with. And we're looking at the differences in what gene pathways or what molecular pathways are being involved that make these different strains respond to stress in a different way. So in fact, what we've done here is we've used heat as a stress. And what we're looking at is survival, where we take the yeast and we put them in very hot water for 7 minutes. And that kills them down 3 orders of magnitude. But if we pre-treat the cells with a mild heat shock of 37 degrees for increasing time out to 60 minutes, you can see with more time at a pre-heat shock, they develop resistance to that high heat killing dose of 7 minutes. So they develop after 60 minutes, you can see that the strains have developed probably 2 orders of magnitude of resistance to heat. And we're studying the molecular pathways and the regulators of that, and asking why are the BioSentinel yeast responding different than the other wild type yeast. We have lots of work to do, and things are going, and hopefully we'll get back into the labs in the next couple of months with these yeast strains. All right, so that's pretty well all we have to say. I just want to acknowledge that a lot of this work was paid for by Bruce Power, funded by our national funding agency, NSERC, and MyTax, which Christine is a postdoc on, and Jake Perkinen, who did a lot of work. SNOLAB has contributed an immense amount of contribution in kind. That incubator you see in the picture on the right at the bottom, the specialized cyst culture incubator, we have no idea what it cost, but I think the last estimate was about $300,000. And that was just basically done for us through the SNOLAB resources. And the final sort of acknowledgement is the NII, which is that little colored symbol above the picture on the right. And that's the Nuclear Innovation Institute, who now are sort of managing Bruce Power's research funding and providing us with the resources to leverage our program. On the left is a picture of one of our grad students, and you can see the guys in the orange are the guys that know what they're doing. The far guy on the right in the orange is Jake Perkins, who's done a lot of work in this project. Dr. Thome, Chris is in the middle there in the orange, and one of our PhD students, Andrew Zarki, is there. When you're underground, you want to make sure you have one of these guys in the orange suits around, and don't go run into the guys with the yellow suits, because the guys with the yellow suits don't know what to do, and that's why you need the orange suits. Again, so we have a lot of people that make this work, and you can see our grad students under my supervision, Chris Thome's supervision, and Suji Theralingham's supervision. These grad students are working very hard on the various aspects of this. We have researchers at NOSM, TC Tai and Simon Lees, who are working on our project. Again, mentioned before, Mark Mendonca is part of this. And of course, the Ames group, the NASA Ames group, Marianne Sola, Sharmila Bhattacharya, and Sergio Santa Maria and Laurel Lindell have been really instrumental in helping us get this done. So with that, I want to thank everybody. Our shark with the laser, thanks everyone. And it would be time to go to the NASA Space Bar. If I was there right now, it's a great place. If you ever get a chance to go to Ames, go to the Space Bar. That's the carpet that you see when you walk in. It's really cool. Thanks very much.
Thanks, Doug. That was fantastic. So we have one question in the chat related to the EAST project. So obviously the radiation environment in space is different than experienced on Earth. Is there any way to analyze effects of the different types of radiation? For example, the contribution of HCE on effects seen in yeast versus gamma and other types of background radiation?
I'll start with that. I think the really cool thing about this yeast system, and it's different than, of course, mammalian systems, is that the yeast system, when they're desiccated, basically has these things in a state of suspended animation, and biologically they're not doing anything. So we can technically take these desiccated yeast that we've set up and got ready for research and take them to various facilities that can give us different radiation qualities and different dose rates. And so we have the ability to irradiate fractionated low doses over many, many weeks or months in our facility. We would like to be able to do super high dose rates and super high doses and low dose rates. There are other facilities we can do protons and alpha particles. And of course, it'd be nice to access the Brookhaven place. And I think NASA's done that already, and I'm pretty sure they have. But once we get a handle on what the background radiation is doing, we should, and some of the mechanisms, then of course, we're going to be looking for different radiation. This is the interesting part about this study, is that I don't think anyone really knows what happens when DNA gets damaged, but does not get immediately repaired or repaired for a long time in a living cell. So, you know, there's free radical reactions that happen very fast in living systems, and repair starts happening right away. And then there's chemical reactions that happen very fast, but we don't know what happens when you protract this and don't do anything with the DNA until eventually you wake them up and say, okay, now how are you going to repair this? So that's a great question. And Chris, you got anything you want to add to that?
No, I'll just say that we've kind of got a pretty good handle now on, in terms of our surface control environment, and then down in SNOLAB, what the contribution is from those different particle types. And so, and I know that they've extensively studied this in space, what those particle types are. And so that is something that we can look at and kind of compare, what the different levels are and the contribution from each one of those particle types.
5 to 10 years from now, what do you hope to see for the repair project in terms of, I know the experiments are difficult to do and take lots of time, but what is your hope for your 5-year vision of what the repair project might show?
Well, and Chris has done a lot more of this work than I have, but even convincing physicists that precancerous cells aren't going to kill everybody underground is sometimes a challenge. So ultimately, we'd like to do the experiments with mice down there, right? Or mammalian system, that we could get more sort of information about higher organisms and multicellular systems. I'm hoping that in 5 years from now that we get enough data that we can get some funding from people that we can take it to another level. Fruit flies, of course, is another great model. And there is a fruit fly project going on down there, not in our lab, because you don't need any specialized equipment for fruit flies. And they're mostly looking at the stress of underground mining. So pressure, dust, noise, stuff like that. We're trying to engage them on that. But ultimately, we would like to sort of, if we had the funding, to expand this into higher organisms with more relevant endpoints for humans.
Chris, what's your kind of 5-year viewpoint?
Yeah, no, I agree with exactly what Doug said. So, I mean, it seems like we showed a lot of stuff that we're just getting started on, but in reality, it's taken us 5 years to get to this point. Like I showed what the lab looked like in 2015. And so even just getting approvals to bring something like a microscope underground takes weeks or months at a time when you're talking about working in an active mine. And so we're really just now at the point where we're set up to run the kind of most basic experiments using cell culture and C. elegans. And so, like Doug mentioned, it's kind of taken a lot to get us to that point. And so I think over the next several years, if we can kind of show in these simpler model systems that we are seeing an effect, and that really kind of convince SNOLAB and get funding to kind of take the next step to those more complex multicellular systems, because that's really kind of where we need to go with this. And that's where other groups are starting now to go there. But in terms of the sub-background biology, we haven't really gone much further than the single cell organisms.
Christine, you have a nice system down in SNOLAB now. What's your view for the next several years?
So the C. elegans are an interesting model because there's a C. elegans repository in the States. And because the connectome and the genome have been fully mapped, there's a lot of knockout mutations already available. So we can bring in worms that have targeted genes after I finish my research, and hopefully we actually find some targets that are of interest. Then we can bring down those targets. And my goal is to optogenetically modify them so that we can turn on and off the genes and see how they respond to the different environments. Also, if I do well with this, we intend to move on and bring down neural cells, mammalian cells, from both males and females. The worms are hermaphrodites, so that's a fair bit of a difference between a human and these creatures. The neural cells, being males and females, can come from different regions of the brain, which are comparable to human. So that's my future goals. I'm not sure if they match the advisors that are on this chat.
Always have extra projects you should work on.
A hint, hint.
There was questions about different mammalian cell lines down at SNOLAB. Could actually any of you three talk about the real difficulty of doing cell culture down at SNOLAB and how you've overcome many of these things?
Well, I think there are a number of different systems we can use, and I see Edizam has asked about some different cell lines. I'd like to really try the 10T1/2 transformation assay down there and see if that has the same responses as the CGL1 cell transformation assay. And it'd be great if we could collaborate with them on that. It's a lot of work. These are labor-intensive things and takes time to look at transformation. So we could probably go and reproduce all the experiments that have been done in tissue culture before in various labs with mammalian cells. But we're hoping we want to find out if there's going to be a difference first. And so that our main hypothesis is transformation in cancer risk. So we'll stick with those transformation assays initially and in some of these simpler model systems, and hopefully we'll figure something out.
There's a question, I'm just going to summarize it a bit. It's related to the type or LET of radiation and stimulating the effects. So you saw that U-shaped response. So you think if you use very low doses of higher or different LET environments, that U-shaped response in terms of suppression of spontaneous background will be different? Chris or Doug or Christine? I'll
just say quick that Chris showed the graph that Les Redpath did, and that was using low energy X-rays, similar to mammograms. I haven't looked at the literature recently, but I'm sure they've done different LET experiments with that endpoint. I'm not sure, can't really remember the work that Ed did with the 10T1/2 with different LET. But certainly in our yeast experiments, when we looked at LET, we could irradiate yeast with neutrons or gamma rays and look at the different way the cells responded to those different radiation qualities. And it turns out that the higher LET radiations don't seem to induce an adaptive response in the yeast to the same magnitude as the lower LET stuff. And we hypothesize that it's probably single strand breaks and DNA topology changes that cause the stimulation at the low doses. High doses is a whole different story.
Chris, what are your thoughts on LET versus some of your endpoints?
Yeah, so I think what Doug said, that they've kind of looked at some of those LETs, but I think for ours, the big question is going to be radon. And so I haven't seen a whole lot done looking at adaptive responses and stimulatory effects of low doses of radon, and specifically the alphas that you're going to get from that decay. But with our system, that is something that we can look at. And so we have that specialized tissue culture incubator where we can get rid of radon, but we can also introduce radon to that system as well. And so we can look at, starting from basically no radon in that chamber, if we expose them for periods of time to different levels of radon, we can look at these things like adaptive responses with those alpha particles. I know not a lot has been done with radon in terms of this kind of stuff. So that's something unique that we can do underground.
Thank you. There is one question related to doing these types of experiments under the ocean versus doing the experiments in the mine. And Doug, you touched on this, but the difficulties of doing things at depth, I assume, relates to density. That rock, you can get water equivalents of, very, very deep versus the mine work. So can you do these experiments underwater in specialized type of devices? For instance, there's a lot of work on nuclear submarines, right? So
I think that'd be very cool. I mean, I can't see why you wouldn't be able to do it. You could set up the same systems we have set up underground, and just like you said, either submarines or some underwater station. Then you can, look at pressure and all the other things. I wouldn't know. I guess you could. The whole thing would be controlled for radon, right? Because you wouldn't have any in there. You wouldn't be pumping in. Well, pumping
No, that's a good
surface
point,
area you
yeah.
might, but
Yeah.
So yeah, no, I think that's an interesting thought.
Yeah, maybe just to add to that, it's most of these, it's just the amount of shielding you can get. And so when you talk about the different labs around the world, like the photo that I showed had different depths in rock, but usually we talk about it in terms of the meters of water equivalent. So SNOLAB is equivalent to about 6 kilometers of water. So there's no reason you couldn't use water shielding for that. You would just need a lot more than solid rock. But one of the astroparticle experiments that they're doing is down in Antarctica. And so they're using ice as shielding from cosmic radiation. So it is possible to use other types of materials. Just you need to get that same kind of equivalence that you're going to get from 2 kilometers of rock or 6 kilometers of water.
So we're at the hour. I wanted to thank you, Doug, Christine, and Chris for a fantastic presentation today. And we had really interesting questions. I'm going to turn it over to Cassandra to say goodbye. Thanks, everybody.
Thank you, Mark. Look forward to next month's speaker, Dr. Graciela Adriani, presenting organs on chips on June 17th. Thank you, doctors. Thank you for your time and your informative presentation, and have a wonderful night.
Cheers, everybody. Take care.